Hi. Hope you guys are doing all right. Hopefully there you guys have I finally got a coffee. mm. So that is the bacteria kind of in a nutshell and how to control it for now anyway. I just realized that I've been wearing my hat the whole time. Actually, no, that doesn't look good. I'll just wear the hat. Perfect. All right. So now thank you guys for sticking around. There was a bit of a long section there, and now we're on to I am recording this, and we're on to the last bit there. So if you guys have any questions, keep it all up there, and yeah, that's what we got. So let me get this one here. And apologies. Share screen. Share. Go. It's not letting me share. This is what I wanted. Here we go. So this is what we got here. We're going to be talking about viruses, prions, and fungi. And I just put this on here, and I'm going to test you on it, but just to show you there, it's places and dates of particular diseases. Okay, and yeah, you just have a look and see where then this is all in the past 30 to 40 years, so interesting. All these diseases are still around. They just don't show up very often. Let's get into it, shall we? Viruses. Now, I guess maybe I should have started with mushrooms because we're more closely related to mushrooms than bacteria for sure. And I'm just going to close my door here. Hi guys, you guys going downstairs? Thank you. Kids. Yeah. Very good. So, but I decided there because viruses are kind of cool and we're in the middle of a virus pandemic, so I thought I'd start off with viruses. So, a virus is not a cell. It's what we call acellular. Okay, it's not. Because when we think of a cell, I've shown you pictures there of eukaryotic cells, plant cells. I've even shown you pictures there of bacterial cells, and there's a lot going on. You got ribosomes and DNA, and you got all these things and moving and shaking and moving all around, right? Well, not so there with a virus. It's pretty simplistic, and that's why we call these guys acellular. It is made up of genetic material inside of a protein coat. That's basically it. Viruses do not carry on basically any or all metabolic activities. It can't do anything. And what a virus does is you'll see there that it attaches to a cell. Like it's just kind of like a free floating, it doesn't do anything. But it attaches there to a cell. It injects its genetic material into the cell, and it basically commandeers the cell. That human cell was your cell. Not anymore. And once this virus is taken over that cell, this cell is no longer working for you. It's working for the virus. Yeah, nasty stuff. And there's some shots there. Now, again, there's a whole bunch of viruses. There's COVID. There's all these. I'm looking for what's here in the PowerPoint there. Again, you could do 1,000 PhDs on this slide alone. We're not doing that, okay? They can only reproduce inside of a living cell. So this is the true definition of a parasite. Outside the host, a virus is lifeless and often exists as a crystal. So, you know what I mean? Cold doesn't bother this thing. It just crystallizes and comes back whenever it needs to. Okay? And you just have a look here. It's got, we'll get into more of these, but you got the tail, the head, you've got the legs here. We'll get into a little bit more detail, but I'm not too interested in this. This is a bit heavy for where I want to go. If y'all are familiar with viruses, how they reproduce, who their targets are, and how to get rid of them, I would consider that a success in this course. Okay, so little bits of information as far as this goes. I'm not too concerned. It's more of this a picture there for your own understanding. A virus has absolutely zero means of locomotion. Can't move on its own, can't reproduce on its own, can't do anything. And you'll see here, this is the eukaryotic cell there that we're used to. Okay, and you can see Golgi apparatus, you've got ribosomes, you've got endoplasmic reticulum, smooth and rough, and this is your rough ER, endoplasmic reticulum, this is your smooth, mitochondria, there's your nucleus, nucleole, you got, I mean, there's lots going on here. In comparison here, here's a bacteria, okay? And you remember there was a slide that tells you some bacteria, they have flagella, only coming out one strand, coming out one end. Some have multiple strands coming out one end. Another has single strands coming out both ends. That was a good little slide there. I would know that one. And you have a look here at the virus. Pretty small compared to everything. So viruses are tiny acellular particles, whereas bacteria are small independent cells that can reproduce on their own. Eukaryotic cells are more complex and larger, as you can see, because these eukaryotic cells, they complain all the time. No, they contain many organelles. Okay. Yeah, you have a look here. The typical virus structure there. Despite their diversity, all viruses have an outer capsid. Okay, composed of a protein subunit and nucleic acid core. Nucleic acid, that's our DNA. That's their DNA. Okay, so there's your capsid. Okay, and composed of, and the nucleic acid there can be DNA or RNA. Okay, generally for us, DNA is the library. That's the core. That's what we protect in all of our cells. And we as human beings, we use RNA to make flimsy copies of our DNA. And it's this RNA, these flimsy copies, that travel outside of the nucleus of a cell and go to and do its specific functions. Okay, so for us, DNA is that's how we house our genetic information. And if we need to use our genetic information here or there or wherever, the DNA doesn't move. It stays in the nucleus, but they will make a copy of the DNA, and we call this RNA. And it's a flimsy copy of DNA. But you'll see here for viruses, they can have DNA or RNA. Now remember, RNA is a flimsy copy of DNA. So some viruses there, they don't even have DNA. They just have flimsy copies. Okay, they have flimsy copies of this RNA, and that's the only genetic material they have. And guess what? It's all they need. Okay. But please know that virus will have DNA or RNA, not both. Okay. Vaccines that are made from the cell walls of infected and damaged bacteria. So, yeah, how they make vaccines is, they take this because before I get into that, I'm going to show you guys. This is where we're going to be, but I'll get there, okay? This is one of the things that I want you to know. You'll have to know this there for the quiz as well. Just the whole idea of what is happening, and we'll get there. So, coming back to vaccines, I'll come back to that through answering these other questions there. So, basically, for a virus, there's your covering and your core. That's all there is to a virus particle. Your covering there is your capsid. Okay, that's the head there. That's this one right here. There's your capsid. That's housing your genetic material. Okay, and then you've got an inner core, which is your nucleic acid, which is your DNA or RNA, and some proteins. That's it. There's really not much there to a virus. The negative virus influence, the bad things, we've heard all about these. We've been suffering for 2 years because of the negative impact of COVID-19. They caused infections in both plants and animals. Okay, not necessarily the same one. There are viruses for bugs, for specific species, but the problem is that some vaccines can jump, some viruses have the ability to occasionally jump from species to species, and therefore now you look at the chicken flu or the swine flu, these kinds of things, right? They also destroy cells and are responsible for human diseases such as polio, measles, mumps, influenza, hepatitis, ABC, colds, common colds, coronavirus. That's all it is. And HIV, these kinds of things, okay? And here are just some screenshots, just to spicing up your day there. There's your influenza. Be interesting. What kind of bacteria? What kind is it? Not bad. What's going on here? Human papillomavirus, warts. Okay, the HPV. Just to let you guys know, you should be checked for HPV. You should look into the vaccine. Okay, because warts, and I mean just warts on your foot or warts anywhere. Okay. You're talking about this virus right here, and this virus is known, and this is another recent development there in the past 10 years, that the human papillomavirus is a major cause of cancer. Okay, and we just don't, what I mean. Cancer there, leading to women there getting hysterectomies and all these kinds of things. So please make sure that you are aware of HPV. I don't think I have anything there on the quiz, maybe on the test, but just be careful. Go and get yourself checked out. If you don't have HPV, please make sure you go and get the vaccine, because it's out there. It's ready to go. Okay. Rabies virus, HIV, we've spoken about, and influenza right there. Okay. Here are some others. Viral disease in humans, sexually transmitted diseases, HIV, genital warts. That's that warts. That's that HPV. Please, please, please go and get that checked out. If you need to know, because that's something there that HPV just sits there, and then what? You'll have cancer there 25 years from now. Okay, and be aware, because all cancer, we can get most cancers at stage 1. Stage 4, no. Okay, so the whole idea of medicine and your health, no human being has had more control over their health than you have at this point in time in human history. You are so aware. You have all these tools available to make your health better. Medicine has to be preventative, not remedy. And what I mean by that is, medicine, you have to take control and eat right, eat better, get your checkups, be on top of your own health. Okay? Because after, people only go to the doctor after they don't feel well, and by that point in time, it might be too late. Okay? So be on top of it. Childhood diseases there: mumps, measles, chickenpox, German measles, respiratory diseases, influenza, SARS, skin diseases, there's your warts again, fever blisters, shingles, these are getting blisters, okay, it's related there to the chickenpox, okay, and this one here attacks the nerves, so certain nerves will periodically become inflamed, and you'll get these blisters over that area. Very, very, very not a nice one at all. Digestive tract, gastroenteritis, diarrhea, nervous system. Okay, shingles should be in here for nervous system as well. And we've got all these here. All right. Positive influence of viruses. Is there really? And, you know, I mean, we've got some good pictures coming, and we're going to get some, there's some pictures there of virus there. Certain viruses are used in the control of insect pests. And I think what they've done now is, it was highly controversial. Geez, I don't even know what, it must have been last year. And they released all of these genetically engineered mosquitoes into the Everglades in Florida to try and stop the invasion of future mosquitoes, because the mosquitoes, they're carrying all these different diseases, and but so they'll use viruses there to control a species, maybe eliminate a species. But as much as we hate mosquitoes, if we eliminate all the mosquitoes on Earth, then the entire food chain would just fall apart, because mosquitoes feed billions of animals every day. Bats, and so if there's no mosquitoes, there's no bats, and then if there's no bats, there's no what eats bats, wolves, and the whole food chain falls apart. So the I have concerns about genetically engineering animals and then releasing them out in the wild, because we do not know the impacts of them, and we will not know the impacts of them until it's too late. And yeah, we just it's a crapshoot at best. You look at the down in New Orleans there in the, I think in the 60s or 70s there, that's where the Mississippi River Delta, the Mississippi travels from basically Canada down to the Gulf of Mexico, and they had all these open water fish cages where they'd keep fish, and they brought these carp over from Asia to clean the bottoms of these open water cages. Well, you know what I mean? Every 12 years, the Mississippi floods. So the cages that were this high, now the water's 10 feet above, so everything's out of the cages. So all these carp are out of the cages, and they're not from here. And that's the damage they can do. And it's just Jurassic Park all over again. But they're doing it anyway. What are you going to do? There's your genetic research. Scientists have been able to use viruses to biologically control caterpillars of the European pine sawfly, and the gypsy moth. Yes, and they're actually looking at doing that there for the gypsy moth there and the pine beetle over in British Columbia, because entire forests, as far as you could see, hundreds of kilometers, are dead because of this pine beetle. But this is a good little slide here. The virus life cycle. We've got 4 parts here, but I'm going to go over this one right here. I like these 5 here. Okay, and the virus is attaching. Okay, and this is what happens right here. So there is an attachment. All right, and inside this the cells here, they have these little proteins that stick out. And like HIV is programmed there to attach to the proteins there on a white blood cell. Okay, so there's an attachment right there. That's number 1. Why does it do that? Stop asking me to unmute. I just want to move this around. There we go. So, and then you'll see there now, entry. This thing starts to move down and gravitate in. Okay, sometimes there it will inject its genetic material in, as in this case here. Okay, I'll get to that slide. It's kind of the same slide. All right, so there's attachment, entry, and then we have what's called reverse transcription. So the DNA or genetic RNA coming from the virus there, it's integrated into the cell. Okay, it starts to commandeer, it's getting this stuff ready to go to take over the cell. Okay, and then we've got biosynthesis, where now the cell is working for us. And us, I mean the virus. Sorry, Sorry. At this point in time now, the virus is beginning to work for the cell is beginning to work for the virus. Okay, and then you have maturation, okay, and then you have release. All right, so this is what we're talking about right here. And this is how all viruses do it to some extent. And you have a look at this one right here. This one here only has 4 points. Okay, but how do viruses work? You'll see there, if a host cell is a bacteria, then this virus here is what we call a bacteriophage. A bacteriophage is a bacteria virus. It's a virus that only invades bacteria. So this is a bacteriophage virus. And you'll see there, it injects its genetic material into the bacteria. Okay, so the viral genome enters the host. So in this case here, the virus itself didn't come in. The genetic material came in. Here, it came in. And then you'll see there's your host cell DNA. And what happens here, the viral genome is replicated and transcribed. So right now, this virus is copying its genetic material as mRNA. This mRNA then gets replicated and made into proteins that are only for the virus. The cell isn't working for the body anymore. The cell is now working for the virus. And then, there would be some time here, where maturation is involved, because these things got to get together. Because all these separate little pieces, they all have to come together. Right? So there is kind of a maturation process here, and then we've got the release. Okay? What we've got here, and I'm not going to test you guys on this, but I just wanted to show you guys there about how a mutation works. And mutations happen through exposure. And people not getting their vaccine, you're allowing the spread of this, of any disease to happen, and the more a disease is spreading, the more it's replicating, the more chance of genetic variability, which leads to the more chance of a mutation. Mutations happen all the time, and they can be bad mutations where the individual doesn't survive birth, or dies very early in life. But it could also be a very successful mutation, okay, where it ends up taking over. And so it is now the dominant mutation. You look at the Delta virus right now. Any virus that's out there right now is Delta. It's taken over everything. And you see here we have the whole idea of a human virus and a let's say a chicken virus. They can and will create a mutation. So now that this bird flu virus, which only went after birds, now it can go after us. See, it's got the receptor sites on there so that it can bind with those little proteins there on the outside of a cell. Okay? Before, these proteins could only bind to chicken cells or bird cells. But now, they've got another way in. Infectious disease is classified as an epidemic. We've been using these words epidemic and pandemic throughout the pandemic. All right. But what do they mean? What's going on here? An infectious disease is classified as an epidemic if there are more cases than expected in a certain area for a certain period of time. So an epidemic, you can have an epidemic of 2 people. If today in Vaughan, there are 2 or 3 people who get the Black Plague, well, you've got an epidemic. Because myself as a health researcher, a doctor, I didn't expect any Black Plague there today. So, we've got 3 cases, that's an epidemic. A few cases of a very rare disease may constitute an epidemic, whereas a larger number of very common diseases may not. An outbreak, an epidemic confined to a local area, that is in the 2013-2014 Ebola outbreak. Okay? Global epidemics are called pandemics. And you've got COVID-19 in here as well. Okay? Nothing too crazy there. I think there's one question in there on that, but nothing too crazy, okay? And we're moving on. We are on the tail end of this, I promise. It's a long one, I know. Prions. Now, these ones here are very strange. As much as viruses aren't alive, and we don't really consider them alive, because to be alive, remember your characteristics of life, right? You got to have this and this and this and this and this and this. Well, viruses ain't got any of it, so I guess they're not alive. I don't know what. We're still trying to figure that one out. But because they have DNA and RNA, there is something in there, right? And but prions, these ones here, they don't even have DNA. They don't have RNA. They got nothing. They're just a protein. That's it. It's an infectious protein particle. It's a totally new kind of mechanism of disease. You see, like with the virus here, right? You've got, you've got attachment, entry, okay? You're making your own DNA in here. Then the cell starts making proteins for you, and then, the whole process. Well, this one here, this is a weird one, okay? There is no DNA to inject. There's no RNA. It's just a protein. And this is a protein that causes bad stuff right here. And let's just read it here. It causes a degenerative disease of the nervous system. And these prions here, we call them proteinaceous infectious particle. Protein, okay, so it's just a protein. Proteinaceous, infectious. It is infectious, and it's particle. So normal forms of protein fold into abnormal shapes called prion. So you remember we were talking about there, the proteins there, they get these, when your body's making them, proteins for any type of use, hemoglobin, this and that, they fold and shape, the ribosomes make one amino acid, one, and then you get this big long chain of amino acids, which then ends up folding into this awesome three-dimensional shape. And there's your protein. Perfect. What'll happen is normal folds of protein fold into abnormal shapes. Why they do that? Still kind of on the out and out. Okay? And these abnormal shaped proteins that aren't they're not meant for any type of bodily function. Okay? Like the digestion process, we produce, there's an enzyme in our mouth called amylase. Okay? And I recommend it. It's kind of gross, but if you take a piece of bread, chew it. Not the whole piece of bread, but chew a couple bites. Don't swallow it. Chew it. And chew it for 5 minutes. It'll turn into this disgusting slurry. But what'll happen after about 4 or 5 minutes is this piece of bread will totally go sweet. It'll taste like you just ate a whole bunch of sugar. Why? Because bread is sugar. We have an enzyme that's produced in our mouth that goes in and while we're chewing starts degrading this bread into simple sugars, making the digestion process go. And amylase has a certain three-dimensional position. And on the starch molecules on the bread, they have a certain three-dimensional shape. And this amylase fits perfectly. It's designed to do that. And what happens is, and the starch breaks. And this protein goes off and does another job. So that's the role of proteins. It's all tied to the three-dimensional shape. It's called the lock and key scenario. Something alters these proteins and changes them into abnormally shaped proteins that can do a lot of damage. Not even alive, and we call these guys prions. And there's a lot that's still out. Okay, prions are able to change the shape of other proteins. And you look, what are prions? They're proteases, enzymes for, they are these, you guys have heard of mad cow disease? Okay, scientists believe that mad cow and related diseases are caused by these abnormal proteins. Okay, and that's what they are. The brains of affected cows have these sponge-like appearance. Just deadly what it does. And these cows will end up shaking, and they're disoriented, and ultimately there they need to be culled. Okay? Prions are different from bacteria, viruses, or any other thing that we've ever seen before. They're not alive, as far as we know. They don't have any genetic material to pass on. Yeah, they're just, they're just proteins. You remember these three-dimensional shapes here? Okay? And what will happen is you'll have a three-dimensional shape of this protein, and it'll change, and away you go. And now you've got something different and something malignant, and yeah. Once in the body, prions convert normal proteins into the nervous system into abnormal prion shapes. So this is how this thing is actually kind of propagating. It doesn't actually reproduce on its own. It programs there other proteins to get bent out of shape, get abnormally shaped, and that's how it goes here. And the disease progresses to a mad cow or other similar diseases and death, and eventually death. And you guys can go on YouTube and see. I was thinking about putting a video, but it's kind of sad what happens to these cows and to people as well. Watch and see. Effects of mad cow disease there. We call it bovine spongiform encephalopathy. Okay, spongiform there. Okay. And this is a fatal brain disorder that occurs in cattle and is caused by these prions. Okay. In bovine spongiform encephalopathy there, the cows behave strangely and they eventually die. And this is the whole thing when we get into like bioterrorism, the ability there to genetically engineer organisms, these pathogens, man, we don't even see them. And there could be a lethal dose of pathogens right on your desktop. And it's quite scary. First identified by a veterinarian there in England in 1968, scientifically known as BSE. Diseases may have passed to cattle through the feed containing proteins or bone meal, and you know what I mean. It's the like the whole idea there that they add antibiotics there to the animal feeds, which I don't think they do in the European Union, but here they do, and they add a lot of chemicals, and they'll even add meat byproducts and protein ground up animals to feed these things. And it was highly unregulated for years and years, and we've got this now. The connection there between BSE and humans was uncovered in England there in the 1990s when several young people died of this disease, and Creutzfeldt-Jakob disease. And this is the human version of BSE. Okay? And BSE is thought to have come from a similar disease in sheep called scrapie. Don't worry about that. And there's just another shot there of a cow suffering. Okay? Saddens me. How can humans get BSE? Okay, or the human version of BSE? Humans contract the disease by consuming parts of the nervous system or eating meat from the cow that has been infected. Okay, and again, it's called Creutzfeldt-Jakob disease. It's a fatal disease of the nervous system. Okay, CJD is another form of that in humans there, and can affect us and be confirmed through. And this is the problem. You really can only find out about it, especially with cows there, if you do an autopsy. You know what I mean? So, yeah, it's a bad one. The USDA prohibits imports of live animals there from Europe, and yeah, I didn't put this up here for you to memorize, or I'm not going to ask you this on a test. I put this up here just to show you there that the U.S. Drug and Food Administration there, okay, that handles all the food and all these kinds of things. The European Union has their own, Canada has their own, and they're all stringent measures there to keep pathogens out. And I think that with the onset of COVID and the irreversible damage it's done to people's lives, families' lives, they've lost people there that they've loved dearly, jobs, economic ruin. I think that this is going to be a forefront of the next all future governments and their administrations, is how to keep things out, keep things in. I don't know. It's going to be interesting to see what's going to go on. And you can see generally where we're at. I'm not going into heavy duty micro. You don't need to know heavy duty micro to understand these. There's a Fungi movie there. Okay, don't forget about that. I'm not going to play it now because I'll get into copyright issues there with YouTube. But please don't forget about this Fungi movie here that we're going to be talking about there soon. Okay. Where am I here now? I'm just getting lost. That's right. There we go. Now we're on to the last bit there. We got some mushrooms. And the mushrooms are great. They're really tasty. The ones that are edible, the ones that are inedible, they're poisonous, so don't eat them. Please don't go out and pick mushrooms. There's a whole bunch of them that come around in the time of year, but please have somebody there around you who knows mushrooms, because some of them can be deadly and you'll be dead within half an hour. Over 100,000 species. Only about 100 there or so that are really pathogenic there to human beings. Fungi. Here we're talking about mushrooms, and we'll get into what's involved with them. There's this word here that we talked about there earlier, the whole idea of going into a hospital for one infection and coming out with another. Nosocomial. Okay, that very, very, very important word. Okay, and the whole business of medical device reprocessing is directly involved in this, and trying to limit the amount of nosocomial hospital infections there, because people do die, and I think the number there was over 8,000 people are suspected to die every year from infections that they got from the hospital. Oh, and by the way, the billions of dollars, trillions of dollars in this. When I worked at a mushroom farm there a million years ago, we made maitake, we grew maitake, cremini, shiitake mushrooms, billions of dollars, and a lot of fun too. Just some words here. Mycology actually means study of fungus. Seriously, man, there's a garbage truck outside our house on a Saturday. Perfect. And mycoses, diseases caused by fungi. Oh yeah, and there's bad diseases there. The pathophysiology of fungi. Oh, some of them. They are eukaryotic, so we are actually more closely related to fungi than bacteria. Okay, because they're eukaryotic and they have all of the characteristics of karyotic or eukaryotic cells. Okay, most are multicellular. Okay, you remember hyphae? Okay, you remember how it grows on the extension? Okay, you've got those, and then there are some there that are unicellular there that are yeast. Okay, so if it's a hyphae style mushroom, they will all coordinate together to make one big mushroom up top. But unicellular mushrooms, unicellular fungi like yeast, they're just by themselves. One is one, and this guy here, they are distinct. They'll hang out together, but they're all different. Okay. Absorbs nutrients. I like this word. You've seen it a couple times in the PowerPoints there. Okay. Saprozoic, and the key word is sapro. All right. These absorb dead material. So mushrooms, they only go in on dead logs, dead leaves, things that are dead, not dying, dead. Okay, and so what we call saprozoic parasites. Okay, they can be mutualistic. Okay, and mutualistic means that a fungi can work with an algae, and that's how you get lichen on a rock. You know, the green stuff that grows on rocks? That's a symbiotic relationship, a mutualistic relationship between fungi and algae. Okay? They have powerful hydrolytic enzymes that are able to convert carbs into sugars, so they're very good at breaking down things. The cell wall, what makes mushrooms a little different from everybody else is that their cell walls contain chitin. And this is an amino sugar polysaccharide. Okay? And that's chitin. We'll talk more about it there in a second here. But mushrooms, these guys here, they have chitin. Okay? It's all involved in, remember, these are the guys, not these mushrooms for sure, but mushrooms, fungi, these are the ones that produced antibiotics naturally for their own defense. We've just used them and taken their antibiotics for our own defense. What do we got here now? So basically, again, we're kind of throwing a flat stone against still water, just trying to get some ideas here. We're not going too deep. And it's like what I said there before. If you see a mushroom cap, that's the end stage of this mushroom's kind of what's going on. And, you know, before the mushroom cap even goes, this thing's been living here for weeks. Okay? And you'll see you'll have this kind of like microbial network there, mycelia. Okay? This spider web, you know, that's going through and myceliated there. And we talked about that type of growth there, previous slide there about an hour or two ago. Okay? And what happens is they will all come together and kind of act like one. And they will all divert their resources to making this mushroom cap here. And you'll see there the hyphae are in there and down in here. And then these guys here will produce spores. Okay? And those are the seeds kind of that go on. All right? Just a micrograph here of these, mycelium network. Okay? Zoom in. You can see them right there. All right. Here's another great link right here. I'll take you guys to that one there. Yeah. Here you have these aseptae hyphae, and here you have septae hyphae. And you'll see the difference is the septa. This type of fungi here, it has these segments in here. Okay, we call these septa. And it kind of differentiates one cell from another. But you'll see here that even these ones here, even more primitive, there is no septa. So it's all just kind of one. Okay, so we call these asepta hyphae. And these ones here are called septate hyphae. Okay. Yes, there is a great link there. So please know that there's a link here that you should watch. Okay, and there's another one here that you should watch. Okay, I'm not going to play them here because I'll get in trouble with copyright there with YouTube. So, and just to continue on here, this the whole idea of septate hyphae and aseptae or non-septate hyphae there, you'll see there that there's no septate in here. Okay, and we've got them all in here. So multicellular fungus, filamentous fungus, you'll remember that there from the previous slide there we did earlier today. They form hyphae, which may be septate or non-septate hyphae, okay? That's all. And then you've got these ones here, yeast cells there. They're unicellular. Okay, that's what these guys here. Good. Histoplasma capsulatum is a dimorphic fungus that grows in soil exposed to bird feces or bat feces. Okay, right up in here. It can change forms to survive at different temperatures in the outdoors if it typically grows as a mycelium, as shown in the micrograph bottom left. There you go. But when the spores are inhaled, it responds to the high temperature of the body, 37 degrees. And then now you have a really, really, really bad lung infection. It's actually called histoplasmosis. Okay, so that's caused there by this Histoploma capsulatum. Okay, and that's it right here. That's how these things work. And this thing is gunning for you. You know, that's its job. It's supposed to get in here. It loves your lungs because it's warm, it's moist, it's protected. And that's where, and that's, bad stuff. I don't know what my kids are doing there next door, but I don't know. You try and tell them go away, but they never do. And these are just more for pictures. I'm not really going into anything too crazy. I'm not going to test you on Rhizopus stolonifer, okay? But I just wanted to show you guys there. This is more about sporangiophores here. These are just the spores there from the fungi. These images show asexually produced spores. Okay, and then just more, you know what I mean? It's the VR lab. I can't take you into the lab and show you these things, so all I can do is try and show you guys some pictures. And the sporangia grows at the end of the stalk of these guys. There's your hyphae, right? These are your hyphae, and there are your spores there. And these spores there, they grow at the end. And they're white fuzzy, you know, like when you see bread mold. When you leave a piece of bread out on the counter and it gets all that fuzzy on it, that's what these guys are. There's your sporangium right there. So the moldy white fuzz, sporangiums. And you see when you hit them or touch them, they just fall right off, right? Because they're supposed to fall right off, and they stick right to you. All right, so good enough. Let's move on. And there's some more pictures there. You know what I mean? Nothing too crazy in here. Again, I'm not going after these bacteria. The Brightfield micrograph shows the yeast Candida albicans, the causative agent of Candidas, and thrush. Okay, thrush there is a yeast infection there. More commonly found in females. Okay, painful, but that's it right there. Okay. And just to kind of throw this in here, because I found it at the later date, but this is just how the drug-resistant bacteria work. And you have a look here, you know what I mean? You got all these bugs. They're, and the majority of the bugs here are not drug-resistant, right? These are normal hokey-dokey bugs. And then all of a sudden, maybe you expose these bugs there to some antibiotics. And now you got 2 bugs here that are antibiotic resistant, right? And then, and maybe you use a different round of bacteria of antibiotics on them, and now you've wiped out all of these ones, or the majority of them, and look what happens in a day or 2. And that's I wanted to show you this because it's very, very similar to what's going on, not drug resistance as far as viruses go, but this is kind of what's going on with viruses. Okay, where the Delta variant now has just completely taken over, and that's all that there is. And so eventually these ones here aren't going to make it, and there's only going to be these ones here. So it's really neat, and I'm really interested to see where this pandemic goes. I hope it's stopping. I've read a number of reports there that it's on the downturn across the globe. I hope it is. I hope it is. We've lost enough people, and I don't want to lose any more. And yeah, but other than that, that is what we got. I apologize for the length of there today, but that's it. Now, just to recap, okay, Quiz 3 will open up there this afternoon. I'll send an announcement out. It should be around 3 o'clock. Okay, there's no rush. You can look at it. It's all multiple choice. I think there's about 58. There might be somewhere around. I might knock out a few. There should be around 55, 56, 57 questions. Now, you're gonna have 8 days to do it. All right. And I want you guys. You're gonna have to dig through all the PowerPoints, and I mean every single one. Okay. Some of them there are gonna be really easy peasy. Some of them there may make you think a bit. But you don't need a heavy-duty biology background to do these, okay? And that test will open there around 3 o'clock. It'll close next Sunday at midnight. I'll send an announcement out all about this. And you guys can go back, change your answers. The test won't close. You don't have to complete it. That's how it'll go. So if you guys have any issues with that, let me know. But I think that's where we're at. Did I see some? Hello. Okay, very good. Hi. So let me know what questions there do you guys got. Talk to me. How are we doing?
Hey, Ian.
Hello. How's it going?
I want to ask something. If we submit the test in like the next 5 days, right?
Yeah.
Before the due date, we don't get an extra shot at it, right?
No, there is no shot. There's just one shot, and I'll just leave it open, and you should be able to walk away from it and come back again. And but yeah, I want you guys to. We'll see how it goes. You know what I mean? I've given enough tests in my life, but I thought I'd be more open on this one, and I want to see how you guys go, because a pass is 70% in this course, right? A pass ain't 50. Pass is 70%. So I want to give you guys as much. There's a lot of and you look at today, man. There's more information there that it's crazy. So before you guys do your quiz, have a look at there today. And but yeah, the quiz. There's one shot at it, one crack at it, but it shouldn't close. Everything should be good. You should be able to go back and change answers, go and look and this and that. Because I want you guys to all get high marks. I want you guys to rip this test apart, because this quiz will look exactly the same as your test coming in 2 weeks, but the test you will have to complete in 3 hours. Okay? So that's where we're at right now. So I've given you guys a lot of questions, but you guys have 8 days. You can take your time, putter around, talk to each other, do whatever you guys need to do. You guys can even cheat. You can. You could have somebody there who's got a master's degree in microbiology do this. But even some of those questions you couldn't even do. But the thing is, I want to prepare you guys there for the test in 2 weeks. Because the test, we will have it from 9 o'clock in the morning till 12. For my afternoon class, we will have test 1. It will start at 1 o'clock and it'll finish at 4. So will it take you 3 hours? No. Should probably take you about an hour and 15. But that's where we're at right now. You guys got any more questions there for me? Whew.
Oh yeah, Ian.
Yeah.
All the tests are going to be on the assignment part, right? You just click on assignments and it's there, right?
Oh yeah. Well, here I'll take you right there. I'll just screenshot. I hope I'm still. Yes, I think I'm still screen sharing. Where am I here? That's what I wanted to. So you'll see here that this is my. I'll just take a look at R here. So this is R. This is my other group and my afternoon group. How are you? Good to see y'all. And you see here are your weeklies. This is what mine looks like. But it'll be an assignment and test. And you see it'll look like this. Quiz 1. They're already there, ready to go. And you would just click on this. And there it is right there. And you could do the test. It should never close. It'll be there all week. And that's what we're looking for there. How was that? Did I answer your question? Talk to me.
Yeah, you answered my question.
All right. Thanks, Jonathan. How are we doing? Anyone else? So I don't want you guys to worry. The test will be open. Have some fun with it. Do your best. And it's all. And I will be in contact with you guys. I'll be sending an announcement out. It's not like next week is a holiday, but it's not like we're going to be taking a holiday. Basically, there are no holidays anymore. So I'll be in contact. And if there's any issues there that irritate you, just let me know. Everything's good. How are we doing? Patrick.
How many times can we go back and forth to the quiz?
You should be able to go back all the time. You should be able to go back. That's what I'm hoping. And because I know that there is a feature on Blackboard that won't allow you to go back. So once you finish the question, that's it. No, that's where we're at there. So that's that. Don't leave this to the last end because there are some questions there that could potentially trip you up a little bit. But is there a submit? Yeah, there will be a submit button. So if you've submitted your quiz, be warned, it is submitted. Okay? So if you submit it before it's done, there is nothing I can do because, and please, you can, I've got 2 classes of all these students. I'll leave the test open, but if you hit submit, then that's it. Okay? Because I cannot and I will not unsubmit it. Because if I do that for one person, then now I've got to give everybody a second chance. So please be careful on that. Do not, just be careful, okay? And thanks, Patrick. I appreciate it. Lemuel, I hope you're all right. Anna Q, you guys are awesome. I got nothing. If you guys got nothing, get out of here. Enjoy the rest of your day. Oh, you guys have another class to go to. And let me know if there's any... Oh, and thank you guys for letting me know about technical issues. If there is a technical issue or something there you don't understand, let me know because you guys are my eyes and ears out there. Thank you, guys. Thank you. Have a great weekend. I'll post this there to YouTube in a little bit. Thanks, Monica. You're great. Marilyn, keep on trucking, man. You guys are doing good. Get out of here. I'm closing her down, and I'll send you guys an announcement about quiz 1 there. It'll be open there in a couple of hours. C'est bon? Bye, guys. Have a great week and happy Thanksgiving.